Welcome to the Deep Dive, where we take a deep dive into some really cool science news.
Yeah.
We've got some pretty fascinating research in genomics to cover today.
Oh, definitely.
Articles from Nature, News Medical, and the RNA-seq blog.
All pointing to some really incredible advancements, yeah.
Okay, so there's this study that's been making some big waves in the aging research community.
Oh, yeah.
Scientists at UC San Diego, they found a link between genetic mutations and these epigenetic modifications.
Right, in the aging process.
You know, how we age.
This is big.
Yeah.
Huge, actually.
Why is that?
Well, it challenges how we've traditionally thought about aging.
Okay.
You know, like it's either random genetic mutations or these predictable epigenetic changes.
Right.
This research is saying it's more like a back and forth.
It's not just about the genes we inherit, then.
No, it's also about how those genes are expressed over time.
So, like, throughout our lives.
Yeah, throughout our lives. The study found that as we age, genetic mutations can actually trigger changes in our cells.
Like in the epigenomes.
Exactly.
Yeah.
Which can affect, which genes are turned on or off.
Which ultimately affects
our bodies. Hey.
So that's kind of like, it's like our genes are telling a story.
Yeah.
And then the epigenetic changes, it's like how that story unfolds.
I like that analogy.
Thanks.
What's really exciting is that this could lead to new anti-aging therapies.
Oh, wow.
Like, imagine if we could target specific epigenetic pathways to slow down aging or even
Reverse it.
reverse it. It's like science fiction becoming reality.
It really is.
All right, let's move on to cancer research.
Another area where we're seeing some huge breakthroughs.
Researchers at Comenius University, they've developed this tool called mFISHseq.
Yeah, mFISHseq.
And this is supposed to be a more precise way to predict how breast cancer patients respond to treatment.
Much more precise.
Okay, so how is it different? What makes it so much more accurate? Well, traditional methods, they just don't capture the complexity of breast cancer.
They don't.
No.
In what way?
Well, they might analyze a limited number of biomarkers, or they don't look at the spatial arrangement of cells within a tumor. mFISHseq gives us a much more complete picture.
So like
looking at the cancer through a much stronger microscope.
Much higher resolution.
Exactly.
So how does it do that?
It combines fluorescence in situ hybridization, or FISH,
Okay.
and RNA sequencing.
I see.
mFISHseq helps us visualize where specific genes are within cells,
Mm-hmm.
and RNA sequencing shows us how active those genes are.
Okay.
By putting those together, mFISHseq can identify even tiny differences in gene expression patterns.
Between different areas of a tumor.
Yes.
This must be so helpful for oncologists.
It is, yeah.
When they're trying to decide the best treatment plan.
Absolutely. It lets them move away from that one-size-fits-all approach.
Right.
And tailor the treatment to each patient.
We're getting closer to this personalized medicine.
We are.
Where treatments are as unique as the people who need them.
Absolutely.
And of course, we can't talk about genomics without talking about artificial intelligence.
Right.
Researchers at Mount Sinai have developed an AI tool called SCORPIO that can predict how well cancer patients will respond to a type of immunotherapy.
Oh, ICI therapy.
ICI therapy, yeah.
Immune checkpoint inhibitor therapy.
It's revolutionized cancer treatment.
It has.
But it doesn't work for everyone.
That's true.
So how does SCORPIO help?
Well, it analyzes blood tests and clinical information to find patterns that are linked with treatment response.
Okay.
Uses these machine learning algorithms to go through tons of data and make really accurate predictions.
That's amazing.
It is.
You can uncover these connections that we might not even be able to see.
Yeah, even the most experienced clinicians might miss them.
Wow.
And what's really cool is that it just needs a simple blood test.
Oh, wow.
No need for biopsies or anything invasive like that.
That's a big deal for patients.
Oh, absolutely.
Both for comfort and...
For early detection.
Early detection, yeah.
Which is huge.
Huge.
Yeah, it could really change the way we treat cancer.
Absolutely.
And speaking of changing how we understand things...
Yeah.
There's the Human Pangenome Reference Sequencing Project.
Oh, right.
Which is working on creating a better map of the human genome.
Like a more complete map.
Yeah, more complete and more representative.
Okay, and that's different from the current reference genome, right?
Exactly. The current one's based on, a limited number of individuals.
Ah, so it's not really a true representation of everyone.
Right, and that can be a problem.
In what way?
Well, it can create blind spots when we're studying diverse populations.
I see.
And it can lead to, disparities in healthcare.
That makes sense.
So this Pangenome project,
Yeah.
it's trying to fix that.
By including data from people of different backgrounds.
Exactly, different backgrounds and ancestries.
It's more inclusive.
Much more.
Okay, so what does this mean for our understanding of human genetics?
Oh, it's huge.
Yeah.
It means we'll have a much more accurate picture of human genetic variation. And that has huge implications for developing new diagnostic tools.
Right.
And treatments that are tailored to each person's genetic makeup.
Personalized medicine.
Exactly, precision medicine for everyone.
That's incredible.
And this ties back into AI, too.
Oh, how so?
Think about AI tools like SCORPIO.
Uh-huh.
They rely on data.
Right.
And the more data they have,
The better they get.
exactly, a richer, more diverse pangenome.
Yeah.
It's like giving SCORPIO a super boost.
So it can make even better predictions about treatment and disease risk.
Exactly.
Wow, so it's like a partnership then.
It is
Genomics and AI working together.
working together to revolutionize healthcare.
That's so cool.
And the pace of innovation, it's not slowing down.
Yeah,
Researchers are always coming up with new techniques and tools.
Any examples that you're really excited about?
Oh, definitely. There's this research on tracking changes in individual cells over time.
What do you mean?
Like, imagine you could rewind a cell's life.
Like a cellular movie.
Exactly.
See how it goes from being healthy to diseased.
Yes. And scientists are developing ways to do that.
Really?
Yeah. With things like single-cell RNA sequencing,
Wow.
we can see how genes are acting in individual cells at different points in time.
It's like having a time-traveling microscope.
It really is. And speaking of looking back in time,
Yeah.
researchers are also making huge progress in understanding the very earliest stages of human development.
Embryonic development.
Exactly. By studying gene regulation during that time, we're learning so much about what makes us who we are.
That's incredible. This research must be so valuable for understanding developmental disorders.
It is. And for improving reproductive health, making sure pregnancies are healthy.
And to handle all of this data,
Yeah.
we need the computing power to keep up, right?
Oh, absolutely.
And that's where companies like Illumina and NVIDIA come in.
Yeah, they're teaming up to accelerate genomic analysis.
Using GPUs.
Yes, those are really good at processing lots of data quickly.
Perfect for genomics.
Exactly.
It's like a match made in heaven.
It is. It'll really speed things up
So researchers can make discoveries faster.
and turn those discoveries into new treatments more quickly.
That's fantastic.
But of course, we have to think about the ethics, too.
Oh, absolutely. With genomics having such a big impact on healthcare,
Right.
what are some of the ethical challenges we need to be aware of?
One of the biggest is data privacy and security.
Makes sense. Genomic data is really personal,
Very personal and sensitive.
so we have to make sure it's protected.
Absolutely. We need to make sure people have control over how their data is used.
And make sure no one misuses it.
Exactly. It's all about building trust.
Trust and responsible development.
Right. And we also need to be careful about biases.
Biases in the data.
Yeah, in the data and in the algorithms.
How so?
Well, if the data used to train AI isn't representative of everyone, it can lead to inaccurate predictions.
Oh, I see.
And that can actually make health disparities worse.
We don't want that.
No, we need to make sure genomic technologies are equitable and inclusive.
For everyone.
Yes, for everyone.
It's a lot to consider.
It is.
So much potential,
Yeah,
but also these potential problems.
we have to be careful.
So how do we move forward? How do we make sure we're using genomics responsibly?
I think the key is open communication.
Communication.
Yeah, we need to talk about this stuff.
Talk about it with who?
With everyone. The public, policymakers, scientists.
Okay.
We need to have these conversations about the ethical side of things.
And the social implications.
Yes, and the legal implications, too.
So it's bigger than just the science.
It is.
It's about how we use the science.
To make the world a better place.
To benefit everyone.
Everyone should have access to these technologies.
Everyone should benefit from them.
Absolutely.
Wow, this has been a really fascinating deep dive into genomics.
It has.
It feels like we're on the verge of something huge, like a paradigm shift in healthcare.
It's a transformation.
Genomics is changing everything.
How we understand human health,
Disease?
even our own identity.
It's exciting
It is.
and a little bit scary.
Yeah.
So much power in this knowledge.
We have to use it wisely.
With compassion.
Absolutely.
Well, thank you for taking us on this journey.
It's been my pleasure.
To our listeners, thanks for tuning in.
Keep those minds curious.
We'll be back soon with another deep dive.
Into the amazing world of science.
Until then, stay curious and keep exploring.